Good afternoon, everyone. Let us continue from where we left yesterday. And yesterday we discussed about the monohybrid cross and the laws of the Mendel. Okay. And when we talk about the monohybrid cross and the laws of Mendel, we talk about two different type of laws we talk about. One was the law of dominance we talked about. Okay. So one was when I'm talking about the monohybrid cross, law of dominance comes into the picture. So what does the law of dominance says? It says that in an individual, the F1 generation, the character that is visible in the F1 generation is a dominant character, and the character that's hidden in the F1 generation is actually a recessive character. And the second one is the law of segregation, right? So what does the segregation mean? Segregation means that when a gamete is being formed, all the gametes are segregating, they're separating from each other, and they then segregate and then give rise to, after the fertilization process, the zygote. Okay, this we have done till yesterday. So today we're going to discuss about a different law and a different pattern of crosses we'll talk about. And this crosses and the pattern we talk about now will be a dihybrid cross we'll talk about. Okay. Now, what is a dihybrid cross? What is it? Is a cross in which we talk about. There is in front of you. Okay. So make sure that when I'm discussing the next law, I will be using a dihybrid cross. What is the meaning of the word dihybrid? It means that the two characters, right? We are taking, like in the earlier crosses, we are taking just one character at a time, like height of a plant we are taking. In a dihybrid cross, I'm taking two characters at one time. Like for example, round and yellow, right? So round is the structure of the leaf and yellow is the color of the leaf. So now you understood we are taking two characters together, and that is how we make a dihybrid cross leaf. So what is a parental generation? It's a true breeding, and it is a RR and YY, means a round and a yellow is there. Another one is wrinkled and the green is there, so you have small r and small y. Now, law of segregation, what does it say? The formation of the gametes, you have two gametes form, R and Y, capital Y. And wrinkled is going to show you small r and small y. The F1 generation is going to show you both the dominant characters, that is round and the yellow is visible. Till here, I think the things are there in your mind, it's clear in your mind. After that, what happens? Now, I'll make a Punnett square and explain you what's exactly happening. So when I'm taking this RR, YY, and this is getting crossed with the recessive one, that is RR and small y, okay? So the F1 generation you are getting is this. F1 generation you're getting is this. This F1 generation is going to be crossed and you'll get a Punnett square, okay? So by crossing the F1 generation, when I'm making a Punnett square, I'm making, I'll not make a complete Punnett square, it's going to take too much of time. But just for you to understand the things, okay? We make a Punnett square after forming the gamete for the F2 generation, the dihybrid cross, okay? So you have the RY over here. You have R and small y over here. You have small r and capital Y over here and small r and small y. In the similar way, in this direction also you're having it. So after doing this whole process of a dihybrid cross, okay, what we are going to come up with in a dihybrid cross, we are going to come up with a ratio of 9:3:3:1. Right? This is, I'm talking about a phenotypic ratio I'm talking about. That is 9:3:3:1, that is, okay? And out of that, 9 is a parental round and yellow. Okay? 1 is again a parental wrinkled and green. What about these 2, 3 and 3 in between? These are actually the hybrids, and these hybrids are actually the ones which show you the next law, and that law is known as the law of segregation. That is known as, sorry, not the law of segregation. That law is known as, the law of independent assortment, right? Why I'm using the word independent assortment for over here? Because in my dihybrid cross, okay, if I'm having round and yellow as my parental population, right? There's a combination of a round with the yellow, round color seed with the yellow structure. I might have a hybrid which is actually round but green in color, that is a recessive character, that is okay. Now this is the hybrid I received. How did I receive the hybrid? I received the hybrid because both the characters which were discussed in a dihybrid cross, they have actually independently assorted, right? From there actually comes the law of independent assortment. The ratio of the phenotypic ratio actually explains you the law of independent assortment, where 9 is a parental combination, 1 is again a parental combination. In between 3 and 3, they both are the new combinations. And that is how we say that these are the hybrids are obtained. So we say that both the characters are actually independently assorted. Okay. So I think this is clear in your mind. This is the third law of the Mendel. This is the third law of the Mendel. You need to explain it through a dihybrid cross. Okay. The complete cross can be made. It takes a lot of time, but I've shown you that through this F1 generation, through the F2 generation, this is what you receive. This is the parental population. This is what you receive in a F1 generation, right? Now, after that, you make a Punnett square out of it, and that is how you receive a dihybrid ratio. And this dihybrid ratio explains the law of independent assortment, right? Okay. So this is what there is in front of you. Okay, they have given you the complete cross they have given you, and they have also come up with the same, you can say the combination or the ratio they have come up with. And this ratio is of 9 is to, sorry, I think, right. So this ratio is what? This ratio is of 9 is to 3 is to 3 is to 1. That's in front of, okay? This is 9 is 3 is to 1. Again, you will have the same thing, that our hybrid processes are there, and that is how you receive the hybrid characters. Okay, fine. Now let us come to the next concept which talks about, let us come to the next concept that talks about, right, which talks about the chromosomal inheritance. This talks about a chromosomal inheritance की बातें करता है. What is this chromosomal inheritance? Okay. So till the time when the Mendel's work was discussed, okay, it was clear that the Mendel's work was actually related to the, you can say, he didn't use the word genes or chromosomes at that time. So he said that these are the factors which we are talking about, right? So the chromosomal inheritance, when came to the picture, actually the scientists were able to understand the way in which the Mendel's factors are working, and it is being carried from one generation to another generation. It is the same way in which the chromosomes are getting transferred and the movement of the chromosome during the cell division process. So actually the factors were within the chromosome, and now we know that chromosomes are nothing but the gene. This is known as the chromosomal theory of inheritance, that it is the chromosome that are getting, you can say, inherited, right? And these chromosomes are having inherited. The chromosomes are getting inherited, and this inheritance of the chromosomes are actually happening during the process of the cell division. And the same work which the Mendel did long back, okay, it also presumed that he said that these are the factors. So in the chromosomal theory of inheritance, we now presume that these factors are the part of the chromosomes, and that is how you actually have the chromosomes running in the parallelly to how the Mendel's work was done. Okay. Now, the work of the Mendel was interestingly more of a theoretical work. It was, right. He was doing the experiment and he was putting all the theory, but as such, there were no very proper evidence for those works. Also, Mendel, for the first time, actually, Mendel has started using the mathematics to explain some kind of a biological phenomena. So it was also difficult for a people to apprehend this thing, that how is it possible to understand any biological phenomena with the help of a mathematics? Because during that time, mathematics and the bio were extremely two different, very different, different. You can say there's the field of the very different field of expressions they were. So you have two different. you were having two very different type of these backgrounds. And so first thing was that number thing was that why Mendel's work was not very much published in a wider way, because obviously in that time I'm talking about the year 1865. Okay, so during that time communication was not very proper, and therefore the Mendel's work was not widely spread out in different regions. Number one thing. The second thing was that the second reason why the Mendel's work was required to be once again published was that the genes or factors, okay, in Mendel's work, it was like a stable and a discrete unit, okay, and they do not blend with each other. So obviously, the blending of the gene according to the Mendel is not possible, but most of the scientists not ready to understand this thing. How is it possible that we say that a blending of the pair of the genes are not possible? That's not the right thing to say, okay. Third thing is that the Mendel's use of the mathematical calculations to explain some kind of a biological phenomena, okay, was again not acceptable during that proper time. So obviously, whole of the Mendel work was restudied and rediscovered, and it was in the year 1900. You should remember the name of the three scientists. The De Vries was there, Correns, and the Teschmark. Okay, these were the three scientists who not in a group, actually independently. They rediscovered the Mendel's work and inheritance, and they actually came up with this concept of the chromosomes and how the chromosomes are actually working and how the chromosomes are actually carrying the genes, and they named that factor or a structure as a chromosome. Okay, so by the time in the year 1902, the chromosome movement in the meiosis was worked out, and Sutton and Boveri was able to relate. Now, sometimes the question comes like this also, that they give the name of the scientist and they want you to explain the role of the scientist in that particular field. Okay, so in the year 1902, the movement of the chromosomes were and happening during the meiosis process, that was finally discovered, and this movement of the chromosome was actually parallel to the behavior of the genes or the factors which the Mendel has discussed. Okay, so in order to explain the Mendel's work, it was very important to explain the behavior of the chromosomes. Okay, and obviously this is very important, that actually these chromosomes are having the genes, and these genes on the chromosomes are present in the form of the alleles, and these pair of genes called the alleles later on. So how are they present? This is a chromosome, two arms of the chromosome. They have two genes present, maybe a capital T over here and a small t over this side. So always in a chromosome, the genes are present in a pair form, and that is how the genes are located on these homologous sites on the homologous chromosomes. So whenever it has been asked that how the Mendel's, number one thing, why it was required to study the Mendel's work or to rediscover the Mendel's work, number one point, and who were the scientists who contributed to it. So initially you need to write about the De Vries, and then you have Correns and Teschmark. They initially rediscovered the Mendel's work. Later on, the work on the chromosome was given up by the Sutton and the Boveri, and they actually explained that the movement of the genes or factor as happening in the chromosome is also happening in the genes also, and that can be related to the work that was referred by the Mendel. Fine. So make sure that, and this is actually whatever the Sutton gave. The Sutton gave an argument of the pairing of the genes and all those things. Okay, this is all actually the pairing and separation of the chromosome during the meiosis process happening. Okay, this is all related to the Mendelian principle, and this later on came to be known as a chromosomal theory of inheritance. Okay. Once this was being done, later on there's some more work related to the Thomas Morgan. Like the Mendel's work was completely plant-based, okay? So Morgan worked on a Drosophila, that's a fruit fly. So Drosophila melanogaster we talk about, he worked on this thing. And he found very suitable for such that they could also grow up a very simple synthetic medium in the laboratory. Because these are very small fruit flies, these are, and they have a lifespan of hardly 1 or 2 weeks are there, and they produce the progenies in very good large number. Okay, that is why the Morgan was able to use Drosophila melanogaster to understand the behavior of the genes, especially in the case of the insects or the animals. Okay, Mendel's work was particularly on the plants, but Morgan's work, for the first time, he worked on the Drosophila melanogaster, that's a fruit fly. Okay, and that is how he was able to show the result in quick successions, because the lifespan of such type of insects are just 1 to 2 weeks. Okay, So he was able to give lot many number of differentiations and the heredity variations that were all possible, and also different little bit of heredity variations of such fruit flies can also be observed under a low power microscope also. So these were the results of why the Drosophila and Melanogaster was used. Okay, so rediscovery of the Mendel's work, again a very very important concept this is, so make sure that you are reading it hard. Okay, fine. Next, we come to the other part of the chapter, which says something different from what the Mendel has discovered. Okay, Mendel has always said that the genes are assorted independently. They have the independent assortment. They are segregated. The gametes are segregated. Okay, he was right in his own way, but there are certain links, genes which are actually linked to each other. Again, the work of the Morgan comes into the picture. And Morgan was able to show to the world that genes are actually able to link themselves and they move together. They actually have a physical association of the two genes, and he named this work as a linkage. Okay. And to describe the physical association genes, chromosome, and the term recombination to describe the generation of the two non-parental combination. Okay. Anything that is not a parental combination becomes a recombination, right? What did Morgan said that there is a physical association of the linkage of the genes. These two genes will always grow together. Morgan and his team has found that even when the genes were grouped on the same chromosome, some genes were tightly linked, okay? And some genes maybe not that tightly linked. I'll show you with the diagram. And this is that given, okay? If you can see cross A and cross B over here, okay? This one, the first one is having a parental type as a 98.7%. And the second time is having the parent type 62.8%. There is more linkage in the first type and there's a more recombination in the second type of a cross. That depends upon the distance between the two genes. If you can see from this diagram, the Y and W genes are very close to each other. On the other hand, W and M gene in the other chromosome is liberated, situated at a very long distance. Okay. So lesser the distance between the two genes, they will be more linked. More linked means always you have a higher parental combination. This you have to understand, and you have to understand this thing that more the linkage is happening of the two genes, the more parental character you're receiving. Okay, so Y and W genes, right? They were very close to each other. So they have this physical associations. Every time, whenever the chromosomes are moving, the genes are moving together. That is why you have around 98.7% of the parental recombination in this parental combination, sorry, in this particular cross. What about the second cross where we discussed about W and the M gene we are talking about? Look at the distance from W and it's quite huge it is. W is on one side of the chromosome, the M is on the other side. Now the distance has increased between W and M. With the increase in this type of a distance, the linkage level goes down, right? And that is why, because the linkage level has gone down. So you have a recombination had gone up, that is around 37%, and parental combination gone down, that is around 62%. Okay. So this is an important thing. The work of the Morgan is important in terms of the linkage. Linkage is also something that is showing an anomaly to the like codominance and incomplete dominance. The work which was given by the Mendel. It is showing some kind of anomaly or a difference in the work that is given by the Mendel. Okay, fine. There are a few more terms are there. I would like to discuss these terms also. A little bit of information regarding these terms are important from your standard point of view. Okay, so like we talk about, one term is a polygenic inheritance, like as a term is representing polygenic, right? Because there is an involvement of multiple genes in for any one character, or it is an influence is there. So we use the word polygenic inheritance. Okay. Like for example, if I talk about maybe the height of an individual or a skin color, if I talk about, suppose it's a combination of A, B, and C gene. Okay. The more number of A's, the more number of B's, the more number of C's will increase the height. Okay, so you have less number of these genes will decrease the height. So it is an additive effect of such genes that's happening. In the same way, if I talk about a skin color, if I talk about, right? In case of a skin color, you are having A, B, C genes are there, suppose. So multiple genes are affecting one character. What is that character? It's a skin color. It is. More the number of A's, more the number of B's, more the number C, the darker the skin color is. Lesser the number of A's, lesser the number of B, lesser the number of C's, it's the lighter the skin color. So what is this actually happening? It is an additive effect of one particular gene, more than one gene, for deciding one character, right? Such type of inheritance is termed as a polygenic inheritance. So the example of the polygenic inheritance you can give is a skin color, the best example you can give. Then there is one more concept of a pleiotropy gene, right? If what is happening is that if a single gene can exhibit multiple phenotypic characters, right? Like for example, they are talking about a disease called a genital disease that is known as phenylketonuria. It is known as, okay. Now this particular disease is due to the problem in the genetic setup which decides the metabolic pathways, right, which occurs in the human. The disease is caused by the mutation of one gene, right, that codes for the enzyme phenylalanine hydroxylase. But what is happening is that this is going to manifest the reduction in the hair and the pigmentation also. So one disease is causing some other two effects also. Did you get my point? One disease is causing some other two effects also. That is the mental retardation and the hair and skin pigmentation. A single gene is able to exhibit more than one character. So that condition is known as a pleiotropy condition. So in a pleiotropy condition, we always give an example of a phenylketonuria, okay? Now, before I go to the next thing, I would like to talk about the pedigree analysis now and few kinds of the genetic disorders. Again, such type of questions are asked in the 3 markers they are asked, okay? So we have the pedigree analysis. Before going to the detail of the pedigree analysis, just a minute. Okay. So for the pedigree analysis, okay, you need to remember there are certain type of, you can say, the symbols are there, okay. Now, if you can look at these symbols in front of you, like the box you're giving male and female and the unspecified sex and the affected individuals. So when you are coloring the individuals, it is showing you the affected individual, and that is how, okay. So pedigree analysis, generally 2 pedigrees are given in your NCERT, and I'll refer to these 2 pedigrees, okay. The first analysis is of a myotonic dystrophy. It is an autosomal dominant disease, right? How do you get to know that it's an autosomal disease? Look at the genes. Look at the individuals who are affected. If something is autosomally controlled, it will be there both in male and the female individuals, okay? So in generation number 1, this is generation number 1, this is generation number 2, and this is generation number 3. So we are talking about an autosomal dominant gene. Every generation it is present, that means it's a dominant character. Also, because it is present equally in the both male and the female individual, that is not a sex-linked chromosome gene. It is actually an autosomal gene. The second which they have given is an again autosomal, but it's a recessive trait it is. So how do you differentiate between a recessive and a dominant trait? The recessive trait is going to skip any one generation. Okay, so now you can see the difference between a pedigree A and pedigree B. The pedigree A is of a trait that is dominant, right? And pedigree B is of a trait that is recessive. Both are actually the ones which are having, you can say, this thing. The ones which are, both of them are the ones which are showing you individuals, both male and female, means they are recessive in nature. But the first one in all the 3 generations, you can see the people are affected with it. Okay, so that is why we say that's a dominant character. In the second case, it is only the second and the third generation that is got affected. But the first generation has skipped that gene. Okay, might be it is present somewhere, but it is a hidden. In that case, we say that it's a recessive gene that is hidden. Okay. Now, let us further go to it and talk about few of the Mendelian disorders also. Right? So few of the Mendelian disorders, if I'm talking about, I'm first talking about color blindness. So we talk about the color blindness as a disease we are talking about. Few of the things you need to remember when you're talking about the disease, the things are not very much in detail, but still you need to remember. Number one thing is that a color blindness is a disease which is a sex-linked recessive disorder. Very important, okay? It's a recessive disorder, but it's a sex-linked recessive disorder. What happens in this gene? What happens during this disorder? A person is not able to distinguish between a red and a green color. Other colors is going to see perfectly. But when we are showing him a green color or a red color, then he get confused whether it's a green or a red color, okay? So color blindness is between red and green color, difficulty to discriminate between a red and a green color, okay? And the reason why this disorder is there is that there is a gene on the X chromosome that gets mutated, right? And because of this mutation of the gene, this defect occurs, and it basically occurs in more of the males it is occurring. Around 8% of the males are affected with it, only 0.5% of the female are affected to it, okay? Red color blindness around the X chromosome, males see. If it is very simple to understand this thing. So because the X chromosomes, males are getting from the mother. So if the mother is a carrier and she gives it to the son, then son becomes a colorblind person. Okay. Obviously, there can be different kind of combinations there are there. But you remember two very important things, that what is color blindness and what is the reason for the color blindness. Okay. Let us come to the second disease. It's again also a sex-linked, a recessive disease. This is okay. Now, this is a disease which is similar to hemophilia, because similar to the color blindness. This is called hemophilia, it is known as, okay. And this hemophilia disease, there is a, you can say, a set of proteins are there that leads to the clotting process. When the blood starts from the open artery or from a injured artery, when blood is coming out, there's a process of 13 reactions that are happening, okay? And that is how it stops the flow of the blood and the clot development happens over there, okay? The person who is hemophilic, in that particular person, it is going to be like one of the proteins out of all those cascade of protein is missing. Obviously, it is going to take more time for that particular person to stop the flow of the blood. It means the clotting time is more in this particular kind of people. Hemophilia is also known as a royal disease because it is there in Queen Victoria's pedigree of Queen Victoria, where most of the descendants, very hard number of descendants, do show the hemophilia, where the Queen Victoria was actually the carrier of the gene. Okay. Sickle cell anemia is again there, again a recessive trait this is. And what happens in a sickle cell anemia, the hemoglobin, the structure of the hemoglobin is changed, okay, because of this faulty HBS gene. And this leads to a change in the structure of the RBC. So instead of the RBC being, you can say, biconcave in nature, instead of the RBC being biconcave in nature, it changed something to this type of a structure, and that is a sickle shape structure. That is a sickle shape structure. Okay, so sickle type of gene is like that, where actually such type of changes are happening. Okay, and such type of RBCs that has changed the shape to the sickle shape, they are not able to carry oxygen with them. So such type of people, they face extreme level of anemia, and at that point, it can be very dangerous for the life of the individual. Okay, so they have given you an example also over here. See, the RBCs over here are the concave shape. But with the faulty hemoglobin gene, it has actually changed the shape to the sickle cell. Okay. Few more diseases they have given. One is the phenylketonuria they have mentioned. Okay. This phenylketonuria is a disease where it's a metabolic disorder disease. It is okay, and but it is due to the absence of an enzyme that converts the amino acid phenylalanine to a tyrosine. Right. happens during the enzymatic reactions happening in your body? The phenylalanine is a kind of an amino acid that is prepared, okay, because protein metabolism is happening in the body. So there is an enzyme that converts this phenylalanine into a safer product that is called the tyrosine. If the phenylalanine is getting accumulated in the body, after some time it gets converted into phenyl pyruvic acid, and this phenyl pyruvic acid reaches the brain of the individual and might lead to some kind of a mental retardation also, and also because it doesn't it is not absorbed by the kidney, so it is sometimes present in the urine also. Okay, so phenylketonuria we talk about, it's known as the inborn error of metabolism, mean right at the time when the child is born, okay, such kind of error in the metabolism is happening inside him because of the faulty gene which is not able to make an enzyme that leads to the conversion of the phenylalanine to the tyrosine. Slowly this phenyl pyruvic acid keeps on accumulating inside the brain, and that is how actually the mental retardation and some sort of diseases are likely to happen. Okay, fine. The next one they have mentioned is about the thalassemia. They have mentioned. Now, the thalassemia is again an autosomal linked gene and recessive gene again it is, okay. And again, this is due to the reduced rate of synthesis of one of the globin alpha and the beta of the hemoglobin, all right? So what is happening is that, see, hemoglobin has got two alpha and the two beta genes chains they are having, right? Proteinaceous chain they are having. So if there is a defect in the formation of the alpha and the beta gene, the abnormal hemoglobin is going to be made, right? And thalassemia jo hota hai, it contain a hemoglobin is affected. Then we have thalassemia can be classified in two ways. It's an alpha thalassemia if alpha gene is affected, and while it is a beta thalassemia if the beta globin gene is affected. Okay. Uh, Basically, जो alpha thalassemia होता है, that is controlled by two genes, HBA and HBA1 and HBA2, that is present on chromosome number 16 of each parent. And is observed that due to mutation or the deletion or some kind of a difference in this gene, the less production of the alpha globin molecules are there, right? That happens in the alpha thalassemia. What happens in a beta thalassemia? Beta thalassemia is controlled by HBB gene that is present on the chromosome number 11. So remember, both the thalassemias are very different, right? The beta thalassemia is controlled by the gene that is present on chromosome number 16, and the beta thalassemia is controlled by the gene that's present on chromosome number 11, right? Thalassemia also differs from the sickle cell anemia. Like the same thing is happening, sickle cell anemia also, that the hemoglobin, faulty hemoglobin is being formed. But remember that thalassemia is a quantitative gene, right? So you can say that quantitatively you have either a very extreme case of a thalassemia or a normal case of a thalassemia, depending upon how many type of genes are faulting, right? So that is how it is different from a sickle. Sickle cell is only one gene that leads to the shape of a sickle cell inside of the RBC. This is the gene that is having a additive effect it is having, right? The thalassemia is a quantitative issue, right? Because two globin genes are being formed, okay? And that can lead to the beta thalassemia or the alpha thalassemia. Again, the level of the thalassemia are very different, right? This particular paragraph is not there in the old NCERT. To make sure that you are referring to the new NCERT to understand this complete concept of the thalassemia. Fine. Okay. So these are the few mental disorders we talked about. We talked about the diseases like hemophilia. We talked about the color blindness, hemophilia, sickle cell anemia, phenylketonuria, and the thalassemia. Fine. Okay. Last topic of this chapter is related to the chromosomal disorders. So chromosomal disorders are the limited in numbers, right? The chromosomal disorders are either the aneuploidy or the polyploidy condition, right? What is the aneuploidy condition? For example, there's a total number of chromosomes in the humans is 23. 23 in the gametes it is 23. So if from 23 it becomes plus 1, 24, or it goes to 1 less, 22 or 21, okay, or there's some extra copy of any chromosome, like in case of the Down syndrome you have extra copy of the 21st chromosome, right? In the Turner syndrome is the absence of one X chromosome in the human female. So such thing, something like that, where the addition or deletion of the chromosomes are there, we say that this condition is an aneuploidy condition. On the other hand, if a complete whole set of chromosomes are altered, that are not present in the human being, maybe present in some plants, okay, that condition is known as a polyploidy condition. That is known. So now you understood the difference between aneuploidy and a polyploidy, where there's a change in only one chromosome number, either one addition or one deletion. We say that the condition is aneuploidy. But if there is a complete set of changes, 4 and 6 and 8 and 10 and that is more prevalent in the case of the plants, okay, then we say that's a polyploidy condition. That's okay, fine. Let us go to some disorders that are there in the humans, okay. There are three they are mentioned in your NCERT. One is the Down syndrome, Klinefelter syndrome, and the Turner syndrome are there, okay. What's the Down syndrome? There is an additional copy of the 21st chromosome, right? So there are how many chromosomes are there? 23 chromosomes are there. So the additional copy of the 21st chromosome, and this was discovered by the Langdon Down. So in order to give honor to this man, the syndrome is known as a Down syndrome, known as, okay. And such particular people, they are very short-heighted people. They have a very small round neck is there, furrowed tongue is there, their mouth slightly open. And important thing is that these type of people are having some kind of a mental retardation also. They have some kind of a psychomotor retardation also, some kind of a physical retardation is also there. So Down syndrome leads to multiple factors. That is why we use the word syndrome for them. How it is produced? The trisomy of the 21st chromosome means the additional set of the 21st chromosome is present, and that leads to all these kind of abnormalities, okay? So Down syndrome. Second one is known as the Klinefelter syndrome, that is known as. In this particular case, you have the additional copy of an X chromosome. So for example, in a human being, it should be XY or XX. So if it is double XY, how many number of chromosomes will be there? It will be a 47. And instead of 46, there will be a 47 number of chromosomes. So with this extra X chromosome makes a person sterile in nature and could be the reason of, again, the mental retardation and some kind of an abnormal characters in the individuals, okay. One more syndrome is discussed over here, and that is called the Turner syndrome, that is known as. So like Klinefelter syndrome was the additional copy of the X chromosome that leads to all kind of abnormalities, okay. Turner syndrome, you have a lesser number of the chromosome. Means instead of having either XY or XX, you are having X and nothing with it. XO we talk about. That is called the XO. So how many number of chromosomes it will be? Just the 45 number of chromosomes will be. So in this 45 number of chromosomes, again, because of the absence of that extra 1X chromosome, there are a lot many abnormalities, sterilities happening in the individuals, and this leads to some kind of a syndrome, and that is known as the Turner syndrome. That is known as, okay. So remember all the disorders which we are talking about, whatever is at least there in the NCERT, you should be knowing about it. So again, I'll go through what we have studied: color blindness, hemophilia, sickle cell anemia. Then we studied about the phenylketonuria, thalassemia. These were all the disorders like mutations we talk about, Mendelian disorders we call them. Okay, then we have the chromosomal. Mendelian disorders related to some kind of a gene. Some abnormality in the gene is there. That's why we call it as a Mendelian disorder. The second disorders are the chromosomal disorders are there. So there we talk about the complete set of chromosomes, or the few chromosomal extra copies are there, or somewhere the copies are not present over there, right? So we call it as a chromosomal disorder. So you have an aneuploidy and a polyploidy are the two conditions. Aneuploidy where the extra number of the chromosome is there, and the polyploidy where the complete set of chromosomes are there, right? So that is how the things are. Then we have Down syndrome, Klinefelter syndromes, and the Turner syndrome as an example. Fine. So this completes our chapter on the principle of inheritance. A very important chapter, holds a good amount of weightage also. Few questions I would like to take right now. We have 5 minutes of time left. So like, for example, question number 9, briefly mention the contribution of the Morgan in the genetics, right? Now, when they're asking you the work of the Morgan, you talk completely of the work he has done on the Drosophila, how he rediscovered the gene and everything, why he chose Drosophila. So that completes the whole thing. So this such type of questions can be of a 3 marker also, okay? Like question number 4, they have said, explain the law of dominance using a monohybrid cross. They have mentioned this question that you have to use a monohybrid cross, but I would suggest that you should be knowing about it. And when you're discussing the law of dominance, when you're discussing the law of segregation, you're using a monohybrid crop. When you're discussing the law of independent assortment, at times you need to discuss the dihybrid cross. So that again, the difference amongst the both should be very well explained by you. Okay. So you have the law of dominance. Then they are talking about the different terminologies also. Again, a good question it is. Explain the following terms with examples. Okay. So in a codominance, you give an example of the blood group AB, where the both A and the B genes are expressing themselves. In the incomplete dominance, you talk about where we say that none of the genes are expressing themselves, like the pink flower of the shoe flower anterium. Okay, so we talked about the incomplete dominance over there, where both the genes are not able to fully express them, right? So what is the chromosomal theory of inheritance? They are asking. Mention any two autosomal genetic disorders with their symptoms. Okay, so make sure that all these questions are most of the questions from this chapter do also come in the case study form also. So you understand you should be reading the question carefully, and that is how you should be taking care of the case studies also. Okay, so I think the chapter is done. All the things are completed. Just go through the chapter of the NCERT once again and thoroughly all the terminologies, examples, and everything. And that is going to definitely make the chapter more, you'll score more marks in the chapter. Okay. Sure. I'll wind up for today. Thank you so much. Okay. Bye bye.